Okay. So I'm going to talk about a framework that we have developed for reconstructing intra-tumor phylogenies. I'm going to mostly skip the introduction because we have heard it now a couple of times. Just let me say that we want to reconstruct this part of the tree or of the evolution. So when we started this project, we got basically inspired by this picture. And this picture is a tree which has been constructed manually, and the clones were assembled by clustering frequencies. And they ended up with 4 clones by the clustering scheme, and then tried to build a tree. And in the end, they saw, and the scheme for building the tree was basically a parent has to have a higher frequency than a successor, and if there are siblings, then the sum of them has to be lower than the parent. Basically, so they ended up with this tree, and then they saw that, and they also had some reads, so it was sequencing data, where 2 mutations would fall on 1 read. So you could actually see by mutual exclusivity if those come from 1 clone or if they come from different clones. And then they basically had to split up one of their clones into actually 3 of them, and they did not know where to attach how much of this clone. So basically by this idea we started this project, and we are going to look at phased data. We are not going to look at frequencies, but we want to look at fully phased data, because I think the phasing by frequency does not work that well, especially if you go in a bit lower frequency ranges. So the idea is that we have a mixed sample, clone sample. We have phased data from it. I'm going to talk about later what that can be. And we do not want to build a tree like this, a classical tree, but a differentiation hierarchy. So basically we want to know how much mass and how big were the clones that were early in the evolution as well. And we want to have a probabilistic method for that. So we use a tree structure Dirichlet process as a clustering prior, as a prior for clustering, that gives us, first of all, a tree structure, and second of all, masses attached to the nodes and the leaves that sum to 1. So it's the perfect setup for clustering, for mixture modeling. We're going to place the data in the nodes, in the internal nodes, as well as the trees, and the nodes and the leaves, and the nodes, the internal nodes and the leaves are going to correspond to clones later. This framework is very flexible, and it has incorporated a transition kernel, which basically gives you gives you prior probabilities on parameters of nodes given their parent nodes. And this, for us, provides us a link to classical phylogeny or to other prior knowledge that we have of the data. For example, if we look at methylation data, which we're going to do later, their back mutation is something that cannot be ruled out and is even fairly common. If we look at single nucleotides variants, back mutation or multiple mutations is something that is relatively rare, and we can sort of try to rule it out in the transition kernel. We do inference by an MCMC scheme, which we sort of adapted from the original author of this clustering prior, and we made it a bit faster, and we have a more effective way of sampling over the topology space, but I'm not going to go into this. We tried to evaluate the performance of this method based on clustering, and as well as summary statistics of the tree topologies, and we see that this method gets better and better and has a higher outperformance with increasing levels of error rates. So now I'm going to show you 2 examples, 2 data sets that we looked at. One data set is from colon cancer, and another one is from leukemia. So the colon cancer data set was bisulfite sequencing of one locus, that's the RX2 locus, that's a 201 base pair locus, and spans 8 CpGs. And so the idea is basically that since these methylations, when they get transferred to daughter cells, have a very high error rate, that this locus basically can act as a molecular clock for us. It's also neutral in this tissue. It's not expressed. The whole region is basically off, so it's a very nice molecular clock. So we we ran our model, and now I'm going to show you 2 examples. And we had multiple samples per tumor, usually 3 to 5 from the left side, 3 to 5 from the right side. And here's one example, and here you can see what is the mass associated with the first level of the tree, the second, the third, and the fourth. We can see in this example that the right side of this tumor looks fairly much the same in terms of tree topology, while the samples from the left side have quite some variance. We can see the same effect if we look at summary statistics from this tree, like the maximum tree depth, as well as the total branch length, that the right side always clusters fairly nicely together, but the left side divides into 2 parts, basically. Another example is from another tumor. Here it's interesting that if we look at summary statistics from these trees, like the total branch length, we can see that the left side and the right side cluster together. It works even better if we use combinations of multiple summary statistics, like the maximum tree depth and the number of clones. We can really see that these 2 sides are separate. So the second example is going to be from a leukemia, and it was single-cell sequencing of whole exome sequencing of 56 cells. There was a very high dropout rate, and about 700 SNVs were identified. And here we just plugged basically a different model into the transition kernel, which encodes for infinite sites model. And the original authors basically had a relatively simple model and concluded from this data that there was a monoclonal evolution. So what do we see? We see also a very strong sort of monoclonal arm of this tree, where there are like 34 plus 7, so 41 cells, which is a good part of the tumor, is associated with this part of the tree. However, we also see that there is quite some branching coming from an early stage of this tumor, which is still seen at the final result of this tumor. So let me conclude. So we have this probabilistic model now for intra-tumor phylogeny reconstruction. Inference, since it's MCMC, is relatively slow and should be faster. But one problem that we ran into and that we haven't solved, and is an open problem, I think, is there is no nice comparison method for these sort of evolutionary trees. So there's no natural sort of distance function or anything that can sort of give a nice score which sort of measures how similar 2 of these trees are. And by that, I want to finish up and acknowledge the people who have contributed. That is Nico here, and Florian Markowitz, and one of his postdocs, Ke Yuan. And thank you for your attention.
Thank you. Chris?
So just about your last comment. So there's a number of methods to compare evolutionary trees. So what is missing in those methods?
So first, our tree, they have masses associated to the nodes. So that's missing. And
Sorry, can you repeat that?
they have masses associated to the nodes. So a node weights, yes. And also we have data inside the tree. All the standard tree methods or phylogenetic trees have all the data at the leaf level.
Yeah, but that should make it simpler.
But we don't have, I don't know, because you have different topologies. You don't know how deep, for example, a specific clone is. You need to measure that, and you need to sort of all compress it into one score, and there's no natural way to do that, I think.
The question here, maybe it's long to answer. At a certain point, you said that you are using some probability construction to gather information about successor from the parent, or vice versa. What is that probability construction that you're using?
It depends on the data set. So one of the data sets, the methylation data set, we basically just had a kernel which said it is relatively unlikely to have mutation forward, but a mutation backward has sort of a similar probability that it can happen. For the SNV data, we did not encode this back mutation, or we had like a much, much lower probability for back mutations.
Just a quick question about the last data set, the single cell data set. Does the tree that you get compare to the method that Nico talked about Monday, if you would run it on that?
Was it the same data set?
We have that paper was earlier than we
Yeah.
ran it with the other method. And there were quite some differences that I don't recall in great detail, but it was not exactly the same.
Okay.
Of course, in this method, as Thomas showed, you cluster together the clones, the sequences explicitly into clones. Whereas in the other approach, we don't do that. So in a way, it's not, we sort of try to compare it, but it's not totally straightforward to compare it, because the vertices of the tree have a different interpretation in the two models.
Yes.
So you have to map them somehow onto each other.
Yeah.
And you have to make some assumptions.
Okay.
Okay, thanks again, Thomas.